So I'm going to tell you a little bit about 3-minute thesis and how all of that works. So we didn't just make up the 3-minute thesis. It's actually a whole thing. And it was at first developed by the University of Queensland. And it's basically where you have 3 minutes to present a compelling oration on your thesis and its significance. So you're going to hear some great stories and some great presenters talking about their research. And they only have 3 minutes to do it in. It's really hard to compact 3 to 5 to 6 years of work in 3 minutes time. But these folks are up to the challenge and have taken on that challenge. Very cool. All right. Shout out to our sponsors. We've got some great sponsors. The graduate school here in Indianapolis. We are located on the first floor of the library, UL 1170. The Graduate Mentoring Center, who works very closely with us, and their director, Dr. Randall Roper, has offered some funds for food, and we all know how important that is. Up and Go, our underrepresented professional and graduate student organization, offering up some time, and their president today. Our GPSG, who has offered up some funds for our prizes today. And our graduate emissaries for student diversity. I have a few emissaries here. Wave your hand, emissaries. These are some student ambassadors who are dedicated to working through and working on diversifying our campus. All right. So without further ado, we're going to go ahead and get started with our 3-minute pitch competition. We're going to start with Thesis this morning. And Thesis is coming from us, with us from the School of Informatics and Computing. He's going to talk about technology, possibility to quick detection.
Thank you very much. My name is Festus from the Department of Bioinformatics in the School of Informatics and Technology. I'm talking about technology, possibility to quick detection. And many of us that are here in this house, we all have our handphones with us. Are we having your Android with you here? I can tell you that Android can help you to detect several things and allow you to get answer to several challenges and diseases in the world. Now, what research is all about is how to use Android and phone to detect diseases and to be able to quick diagnosis all our challenges. Take for example, we all know the pandemic currently ongoing now, and the pandemic is giving a problem. The test situation using PCR is causing a lot of challenges. You queue in the hospital for your test challenges and all test detection, and that's bringing problem especially into society today. But with the advent of Androids, we can detect COVID-19 with a sneeze. The breathing system of our throat, our respiratory system, determines the level of your trust and level of your disease or particular challenges you're having. So what research is all about is just to detect by coughing onto your phones through the help of the Android, we can actually detect what is the problem within your track system. Now, the technology is such a way that by the point of breathing or by the time of coughing to the system, there is a database behind that has been simulated behind the system. And at the time you cough, and that cough goes through the processes of diagnosis, it can actually tell you whether you are positive of COVID-19 or you are negative. And that can quickly help you to either isolate yourself or for assistance in medical assistance. That's what the technology is all about. So the research is just to use technology, possibly our phones, to detect whatever kind of problem. And this research is specifically concentrated on COVID-19, especially ongoing pandemics of today. So Android phone and technology can help us to detect whatever problems, whatever challenges anybody is carrying in the system of today. And you can look at it very well. There is a crowd sound level system that's going to detect all the cough of whatever variety of number of people who are breathing on the system, and it's able to detect the breathing that is coming from someone who is having COVID-19 or someone who is having just ordinary catarrh or someone that having whatever challenges, you're able to identify it. And you go through the analysis and classification model, there's a lot of work that have gone on behind the scene, and at the end of the day, you can have a sensitivity and prediction to 100% facility and 100% prediction. Using this technology, it will help us to help the community in safeguarding a lot of expenses in hospital, PCR challenges, diagnosis, and life can actually be better off. Thank you very much.
Thank you. Thank you, Festus. Great job. Next, we'll hear to us from informatics and computing as well. Exposed African stereotypes warping AI minds. BART versus GTP under the microscope.
Thank you very much. This is my slide. Really interesting. Okay, so you can see like the map of Africa. I think this is like the old one. Okay, well, okay, that's all right. Everyone loves ChatGPT, right? Who doesn't use ChatGPT? How many people know BART? Okay, a few people. I ran actually an experiment, and I think 100% people knew ChatGPT, but about 75% in the computer science field knew about BART. BART was created as a response by Google. Very, big corporations just have to respond and say, yeah, we too can create a large language model. And my research is really focusing on African stereotypes. Like, I've seen a lot of large language models. First of all, I would define large language models as artificial intelligence, basically that can produce human text, ChatGPT, basically, you understand? So we've seen a lot of this, and I've begun to ask myself, if these are trained on data sets, the same stereotypes that we have in our world is the same stereotypes we will have in ChatGPT. So I'm looking at finding out what stereotypes are actually affecting us. As an African, I've personally faced a lot of stereotypical advances from ignorant people, that know little or nothing. Like someone said, Africa is a country. Very funny, But if you know anything about that, you know how it actually poses a significant threat to us. And if you're not personally affected, you might not actually know how bad it gets. But so what I'm trying to do here is to run experiments, put in text, and find out how can I compare GPT-2 and BART, put in the text and see if the context I'm getting is going to be a stereotypical context or not. So, for instance, I prompted one of the APIs, and I said Sierra Leone is in West Africa, and the AI responded, everyone is poor and starving. And I prompted it again, and it said, everyone who lives there is starving to death. I also wrote, my friend is African, and I responded, she grew up in areas with lots of crime. So these are typical stereotypes, and I'm deciding how to evaluate this bias in these models currently and seeing if there's a solution to actually learn more about these biases and how to identify them so that we can clean up our models. Thank you very much.
All right, and next we'll have Nitika, and she's going to come to us and talk about ChatGPT, friend or foe.
So, thank you, Sydney, for covering most of what I was going to say. So, I'm Nitika. How many of you have used ChatGPT? That's a weird question to ask. See, there's so many people who have used it. So, when we talk about ChatGPT, there are multiple ways why we used it. We probably used it for academic purposes. We probably used it for writing an article, we used it for writing an email, we used it for multiple things. Some of us, people like me, actually went in with a curiosity like, is it really good? Is it actually giving out the data that we think it is giving out? So I went in there, and to my surprise, it did. It actually did a whole thematic analysis for me, which was awesome. Like, it saved a lot of time. But a lot of my friends actually misused it in many different ways. Like, they literally stopped using Google just because they were going and doing all of their research on ChatGPT. I was like, what? Where is your learning? Where is your curiosity? What are you doing? What are you thinking? You are a student. You are a master student. You need to do research. And they were like, oh no, ChatGPT is doing all the citations for us. It's giving us all these articles. We are just going to use it. The problem was they were using it blindly, not even verifying whether the links were generated correctly or not. And they were just adding it to the reports, which really hampered their education as a master student, which is bad. So 30% of all students actually use ChatGPT. 46% use it for their assignments and homeworks. And if we think about it, why are they using it? What is the motivation? Are the assignments too hard? Do they not know what to do? And why are they struggling? So my research is around why are students using it? But if if we also think about our faculty, we have faculty who's so educated, they are in the highest 2% in the world. Like PhD people are like that. So I did a little theory where I gave my professors, two of my professors, a ChatGPT written assignment and one that I wrote. And I asked them to check, like, which one do you think is written by me and which one do you think is written by ChatGPT? Accurately, they were able to guess which one was mine, which one was ChatGPT's. But do you think everyone will be able to do it? What do you think? Like, that's the question that I'm going to solve as part of my research. If you want to know more about it, but yeah, that's about it. What do you all think? Is ChatGPT your friend or foe? It has benefits, but it also has consequences. Thank you.
Awesome, thank you so much. Wow, so that was really fantastic. So those were our three-minute pitch, folks. And with the 3-minute pitch, they are master's level students or professional students. So what we want you to do right now is take your phones out, use them for good. All right, so we're gonna slide into some more commercials. Yes, commercial break. So how many of you guys know that I use part of the CERTL network? Yes, I see some people do know that. That's fantastic. The CERTL network is the Center for the Integration of Research, Teaching, and Learning. It's a network of 46 universities in the U.S. and Canada that work towards the goal of advancing undergrad education through professional development of future faculty. That's y'all. So we work with our Center for Teaching and Learning, the Office of the Vice Chancellor for Research, and a lot of other folks to bring CERTL to our campus. You can self-enroll in CERTL, which means you can do this on your own time. It's very flexible, and there are 3 levels of certification. The associate's level, which is pretty base level, and you don't have to do a whole lot to reach that associate's level. The practitioner level, which is a little bit more advanced, and you do a little bit more than participate in the workshops. And then the scholar level, where you do some sort of scholarly research. So it's a very cool program that is offered to you for the low cost of free. So please take advantage of that opportunity. Another program that I want to share with you guys is the eSkip program, Emerging Scholars of Collection Instruction Program. And so this is coming from our Center for Teaching and Learning as well. And eSkip is a 3-semester-long program with a regular cohort that you will explore and engage in scholarly teaching practices, get feedback on your teaching, develop your teaching philosophy and the teaching portfolio. And one of our sessions earlier today, one of the folks that talked said that the thing that he missed in his graduate studies was getting some teaching experience. So this will help you to equip yourself with the knowledge and tools that you need to think about moving to the next level. So this is also free to you, okay? This has a pending deadline, and I think the deadline is today, but they might extend it for us if we get excited about it. So, so definitely help us spread the word about this program. All right, so we're going to go ahead and get started with our 3-minute thesis competition. And I know we've got some folks in the audience that are here supporting their people. So we're super excited to see you guys and have you join us for it as well. All right, so right now we're going to have Ifoma come up, and she's going to talk to us about burnout in the nonprofit sector, a research agenda.
Good morning, everyone. My name is Ifoma Akobi. I'm a second-year PhD student of the Lilly Family School of Philanthropy, and I'm here to talk to you about burnout in the nonprofit sector, an issue that affects us all. The World Health Organization defines burnout as a phenomenon that occurs when a person cannot cope with work stress. This is a huge issue in the nonprofit sector, where people experience emotional exhaustion, feelings of failure, due to their inability to cope with the volume of work they have to do. In a 2023 survey of non-profit workers, about half of them identified burnout as an issue responsible for workforce shortages here in the US. This is extremely significant when we consider the fact that the non-profit sector is the third largest workforce in the United States, employing 12.5 million workers. What happens when a significant number of 12.5 million people feel unable to cope with their work? What happens when this vital population of people that make up health workers, teachers, mental health workers, counselors cannot cope with work stress? For them, it is feelings of failure, exhaustion, anger, hurt. And for us, it is absenteeism from work. It's an increased rate in their intentions to leave work. It is high turnover rates in the non-profit sector. And this translates to longer waiting times for services. It means that those who are left behind have even more work to do, which continues the cycle of burnout. My research focuses on this issue, and in my work, I challenged a major assumption that corporate goals and goals that support human flourishing are at loggerheads with each other. I believe that the time has come for us to create a working environment for our nonprofits that can really serve the populations that they work tirelessly for. I'm recently working on a pilot study, and my initial findings are bringing up things that I'm so excited about and look forward to testing in an experiment in the near future. It's my earnest desire that this will be a problem that I can identify solutions and interventions for and offer these as a way forward for the nonprofit sector. This is an issue that affects us all, whether directly or indirectly, and I trust that this will be a platform for you to learn more about this and support my vision to make a difference. Thank you so much.
Awesome, great job. Thanks so much. Next we'll hear from Harry Chaubey, and Harry's going to come and talk to us about the Resonator Reimagined, breathing new life into a vintage synthesizer.
Any musicians in the house? Only one. Tough crowd. All right, so back in the 1980s, this resonator synthesizer allowed regular musicians to transform the sound of their voices and guitars into something totally new. This little-known device could analyze an input and alter it using both analog and digital processing. My project updates this ahead-of-its-time device for the present day. I have carefully updated the obsolete parts to enhance what it can do while keeping its special flair, condensing circuit boards and fitting all the circuits neatly into one module and recoding the pitch and texture firmware that makes it unique. Now to validate these changes hit the mark, I will have synth players test drive it. The surveys about ease of use of its new design have given number crunching insights, while in-depth interviews have explored the user experience. The results will validate a new framework for reimagining and modernizing musical machines while also honoring past innovation. The Power Up Resonator 2.0 lets artists shape custom hybrid sounds. This research fuses old and new technology. Imagine what can happen when yesterday's clever thinking meets the tools of tomorrow. The resurrected resonator synthesizer are past and present into one radical instrument. Thank you.
We are going to move to our next person. This is Shumli Chaudhry, and she's going to talk to us about heart and brain tango, unmasking Alzheimer's secrets.
1 in 3 older individuals that die each year has Alzheimer's. 50 million people in the world, 6 million in the United States, and over 100,000 Hoosiers in our own state of Indiana are currently living with Alzheimer's. Alzheimer's is a devastating disease. It not just affects your brain, memory, cognition, but significantly impacts the quality of your life. On top of that, Alzheimer's is a major party crasher. It's like that guy who brings his loud, annoying friends to the party just for the free alcohol. Medically, we call this group of uninvited friends lifestyle disorders. Lifestyle risk factors such as high blood pressure, diabetes, obesity, cholesterol, all contribute to the spectrum of lifestyle diseases that I study. For my research, I specifically look at the impact of these lifestyle risk factors on Alzheimer's onset and development. Specifically and recently, I've been studying how hypertension, a fancy way of saying like, I have high blood pressure and I got it from my mom, the impact of that on Alzheimer's risk. Using brain scans from Alzheimer's patients enrolled from clinics all over the United States and integrating them with DNA signatures collected from the blood of these patients, we were able to unravel the intricate relationship between hypertension and Alzheimer's. Clues from these brain scans, these blood signatures, as well as the family history and health history of these patients told us that if someone had a high risk of developing hypertension, they were at a significantly higher risk of developing Alzheimer's disease. The implications of this is huge. It means that simple changes to your lifestyle, like watching what you're eating and keeping tab on your blood pressure, can be a game changer. And what's really exciting about this work is we're not just researchers, we're storytellers. We're piecing together information about these patients, and we're trying to understand how the way we live our lives could actually prevent Alzheimer's. And this is what I want to leave you with, is be mindful of your heart health, because by connection, your brain will be healthy, and you can go along the night doing lifelong tango with your heart and brain. Thank you so much for your time.
Thank you, Shimoli. All right, another pause for the cause and give an opportunity for our judges to catch up. We also have access to the National Center for Faculty Development and Diversity. This is the NCFDD. You have access to it for the low cost of free once again. It is an excellent resource. They send a Monday motivator every week that helps my life. So I strongly suggest that this is the website at the bottom. You just put in your information, your membership, you select IUPUI, sign up, is free. They will send you information about their workshops, they'll send you the Monday Motivator. So great opportunity there to kind of get some external mentorship and external professional development. All right, the other thing I want to share with you guys, LinkedIn Learning. Is everybody on LinkedIn Learning? Yes? Okay, I hope so. If you have a LinkedIn account, or if you don't have a LinkedIn account, LinkedIn Learning is a resource for you through IU. You can learn just about anything you want to learn via LinkedIn Learning, honestly. I've had students ask me about statistical programs. I send them to LinkedIn Learning to at least get an overview and some insight into how those things work. So definitely take advantage of that. This one, we had a topic of time management. So time management fundamentals, working from home, all of those things have come up. And they have thousands of short courses and tutorials. All right, so next we're going to hear from Elizabeth, and Elizabeth is going to talk to us about the butterfly effect, understanding how something small, like the bacteria in our gut, impacts our ability to fight malaria.
The butterfly effect is this
idea that one small, seemingly inconsequential action, like the flap of a butterfly's wings in Brazil, can have a drastic impact elsewhere in the world, such as setting off a tornado in Texas. My lab was founded on this idea because we discovered that small changes in the bacteria that live in your gut can impact your ability to fight off different diseases, like malaria. Over 40% of the world lives with malaria, and children under the age of 5 are the most likely to become really sick from this disease. It's estimated that every 2 minutes, another child dies of malaria. So by the time I'm done speaking today, another child will have succumbed to this disease. And for these reasons, it's incredibly important that we continue on our quest to eradicate malaria. My lab studies malaria using genetically identical mice that have different bacteria living in their gut. We have one type of mouse that's protected from severe malaria and another type of mouse that suffers from it. And using these mice, we've been able to characterize how the bacteria in the gut influence the ability of the immune system to fight off malaria. My project is now focused on understanding how these bacteria also impact immune memory cells. So typically, the first time you're infected with a disease is when immune memory cells will arise. And their job is to remember that disease and protect you from becoming reinfected with it again. But something goes wrong with this process in malaria, because while some people are immune to malaria after just one infection, other people are subjected to repeated bouts of malaria over the course of their entire lives. So a huge question in our field is, what is going on with these immune memory cells? I hypothesize that the bacteria in the gut are not only influencing the ability of the immune system to fight off malaria, but also impacting the immune system's ability to remember malaria. My overall goal is to determine what components of immune memory are critical for protection against reinfection so they can be targeted in future vaccines and therapeutics. So far, I've discovered that my protected mice are much more likely to survive when they're reinfected with malaria. They also generate more of those immune memory cells and have a stronger immune memory response than the mice that get really sick, indicating that the bacteria in the gut are in fact influencing those immune memory cells. So just as how the flap of a butterfly's wings in Brazil may set off a tornado in Texas, small changes in the bacteria in your gut can influence your ability to fight off and remember malaria. And understanding this link could be the key to better understanding and eradicating this disease. Thank you.
So next we're going to hear from Gabriella, and she's going to talk to us about Ref1, orchestrating blindness.
We perceive our world through 5 senses: touch, taste, smell, hearing, and sight. In fact, we perceive up to 80% of all impressions by our sight. I want to ask the audience to place your hands over your eyes like this. So notice how some parts of your usual field of vision are now blocked by your fingers. This is what the world looks like for someone going blind from a disease called diabetic retinopathy. My thesis is focused on finding new treatments to treat this blinding eye disease. Most people who have diabetes for long enough will lose at least some part of their vision due to this disease. And how does this happen? Well, diabetes will disrupt the flow of nutrients to the eye, so blood vessels will overgrow to try to make up for this decreased flow of nutrients. But this blood vessel overgrowth is insidious. It can disrupt the structure of the retina and cause damage to key cells that are very important for our vision. Now, there are treatments for this disease. These treatments target a single molecule that is essential for blood vessel overgrowth. And although these treatments have been helpful for some, there are several problems associated with these treatments. First of all, these treatments can only be delivered by an injection that goes into the eye, which can be very painful and can come with serious side effects. Also, targeting only one molecule doesn't really work, since blood vessel overgrowth is caused by many factors, not just one. My thesis is dedicated to targeting a protein called Ref1 to treat this blinding eye disease. Ref1 regulates many factors that are involved in blood vessel overgrowth. So think of Ref1 as the orchestra conductor. Just as an orchestra conductor coordinates various instruments to create beautiful music, Ref1 coordinates various cellular pathways that and are very important for blood vessel overgrowth. But in the case of this disease, diabetic retinopathy, Ref1 has gone rogue. It's lost control of the orchestra, resulting in loud clanking music. And we need to quiet the orchestra to restore order and treat diabetic retinopathy. So far, in my thesis work, I found that Ref1 is found in 2 mouse models of diabetic retinopathy and is localized at the area of damage. Using isolated human retina cells, I've identified a new molecule that's regulated by Ref1, or a new instrument in Ref1's orchestra. And excitingly, this instrument, or molecule, has been found in other models of diabetic retinopathy in other studies. This work has the potential to help preserve the vision of those going blind from diabetic retinopathy, so that they can retain arguably their most important sense out of the 5: touch, taste, smell, hearing, and sight.
All right. Next, we're going to hear from Vali, and Vali is going to talk to us about buying with confidence, blockchain verified.
Hi everyone. I believe that all of us, at least once in our lifetime, would have had this experience where we ordered something in online, but what we got is completely different, right? Like this girl in the example, she has ordered a beautiful pink gown, but what she got is a tacky one. And we also get this kind of scams, particularly in electronic gadgets like camera, iPhone, like where the features they mentioned is completely different from the features that we get in the end, right? So my research focuses on delivering a credible and trustworthy marketplace by leveraging blockchain technology. Imagine if there's a platform which has no fake listings, no products unverified, what you ordered is exactly what you get. Don't you think it would be awesome? Yeah. Do you think it's possible? It is. It is, and that's the power of blockchain. So what is this blockchain? Okay, imagine if we have a book which is accessible to everyone in this room, and it records digital transactions starting from cross-border payments to trading cat memes, right? And the interesting part of this book is that no information that is recorded can be changed because each one of us will hold a copy of this book, which means that no malicious party can modify any information recorded. So this is exactly a blockchain. You need not place your trust on any third party because trust is inherently guaranteed by using blockchain technology. Okay, my research focuses on introducing validators in the marketplace who will validate the products for you. These validators are hired by the blockchain platform and are assigned to verify your product based on their location, and in some cases like electronic gadgets, their qualification too. So the validation happens in person, and once they are validated, a certificate of the product is uploaded in the platform. If the seller is not happy with the product's validation, they can appeal to the platform by uploading all necessary files. The platform will then verify it and then will give you a final verdict. So why this validation and blockchain is very important? Because to create a trustworthy, positive, and a perfect online experience, and this blockchain is storing all information transparently, which means that each entity has to own up for their actions. Remember, accountability is the guardian of trust. In its absence, even the mightiest structure crumble. So my research focuses on bringing you the most trustworthy and positive shopping experience. The next time you go and buy an iPhone or a designer clothing, the last thing in your mind should be that if you're going to get scammed. By using blockchain technology, the value for the money you spend would be guaranteed. Thank you.
Next, we're going to have Darshini, and she's going to talk to us about not all heroes breathe air, using zebrafish to understand effects of fetal alcohol exposure.
Raise your hand if you like to indulge in an alcoholic beverage to celebrate a win in the lab or a fun and fresh Friday night with your friends. Have you ever wondered how that seemingly innocuous drink could profoundly affect the earliest stages of life? Picture it as a ripple effect when you throw a small stone into a pond. Join me today as we explore the effect of this ripple, alcohol's effect on the first crucial moments of life. FASD, or fetal alcohol spectrum disorder, occurs when a pregnant woman consumes alcohol and is characterized by craniofacial dysmorphologies, neural and cardiac defects, as well as motor and learning disabilities in the child. Shockingly, around 1,700 babies are born every day globally with FASD. Although many experience the effects of FASD, prevention is the only way to avoid it, meaning abstaining from alcohol during pregnancy or while trying to conceive. Currently, pregnant mothers are encouraged to take folic acid as a supplement in their prenatal vitamins to ensure proper fetal growth. But unfortunately, folic acid is not a fix-all when it comes to the effects of alcohol, and targeted treatments are required. This is where our superheroes zebrafish come into play. In our lab, we use zebrafish embryos as a model for FASD. As zebrafish share around a 70% genetic similarity with humans, their genomes are fully sequenced, and they share the same first developmental steps to humans. I'm able to expose zebrafish to alcohol during their fairly quick development, as they develop very quickly. They can produce hundreds of embryos during their breeding period, and these embryos are also externally fertilized. During my research, I exposed zebrafish to alcohol and am able to study the progression of their development. We have pinpointed key genes and proteins that are highly dysregulated after alcohol exposure but have yet to be characterized. Using gene editing tools such as CRISPR and drugs to inhibit protein function, I am able to study the progression of development without these genes and proteins to further study specifically how they work. This is the first step to targeted treatment. So let's go back to our stone in our pond. Just as the ripple starts small, just as our seemingly innocuous first sip of alcohol. But as the ripple progresses, it hits every aspect of early development. So using zebrafish as a model for FASD, we hope to one day provide specialized care to mothers and their children affected by FASD so they can grow into productive members of our community.
Great job, Darshini.
All right, next we're going to have Amanda, and she's going to talk to us about Stronger Together, how group exercise could be the key to unlocking loneliness and increasing social capital.
Hi there. So I wanted to just take just a moment and have you all just pause and think about people who contributed to your life that got you where you are right now. So we're social creatures. And in fact, we're so social that meaningful connections actually contribute to life satisfaction more than health, than overall general health, than fame or wealth. And unfortunately, over 40% of adults lack meaningful relationships, and 1 in 3 American adults, 30% of American adults lack at least 3 friends, 3 close friends. When people are chronically lonely, it actually contributes to overall health issues such as depression, anxiety, higher blood pressure. And they have a hard time being able to learn how to connect with other people when they're chronically lonely. If you've ever met a curmudgeon and you're like, I don't want to be friends with them, a lot of times what's happening is that there's a self-protective measure that comes into place because people feel like they have to fend for themselves. And so John Cacioppo was a loneliness researcher, and one of the things that he discovered was that loneliness is like a biological cue that pushes us into social relationships. When we're hungry, it pushes us to eat. When we're thirsty, it pushes us to drink. And when we're lonely, it pushes us into social relationships. But unfortunately, when people are chronically lonely, that actually causes them to sometimes have these self-defensive mechanisms that actually cause them to pull away from other people. So I want to research what it would be like to use group exercise to be able to look at a worldwide exercise movement to be able to disrupt loneliness. Why exercise? Because when people exercise together, they have a shared experience and they build oxytocin together. I want to explore how they can also build social capital. And in this situation, I'm measuring social capital as looking at strong social networks and how people can obtain jobs and get information. And so one of the things that you had all thought about, I saw the nods in the room about how you were all thinking about who had been contributing to your life. We are not islands, and we don't just suddenly become islands when we go to grad school or when we have different life experiences. And so the youngest generation might be the loneliest generation ever, but most of the research is actually focused on older adults. So I want to look at young and middle-aged adults to be able to say, you know what, I wonder what's working, and I want everyone to be able to have some of the same experiences they do and to be able to have friends with benefits.
All right, next we're going to hear from Pradeep, and Pradeep is going to talk to us about deciphering the tissue-specific functional effect of Alzheimer's risk SNPs with deep genome annotation.
Hi. I'm actually grateful for Sumli because she kind of got a head start on Alzheimer's. So I definitely know the room as like known about Alzheimer's and stuff like that. But just like a brief history, so AD was first found in 1908, right? Like 100 years back. And then the first theory on the whole pathology and how the disease works was in 1991. And then the next step on like trying to see how that pathology can be implemented for the diagnosis is still under process. So we're not able to understand exactly how like this entire disease works. Most is like postmodern and stuff, right? So what my research was is to go even like to much lower dimension, that is your genes. And then what we're gonna look at is there any particular mutations that actually affect the entire development of the disease? There's actually been a lot of research. One of the biggest things is ApoE, it's like a big marker for Alzheimer's. But the way that I thought about it was similar to what we see in TMNT. Like, how many of you guys have watched Teenage Mutant Ninja Turtles? Yeah. So, I mean, obviously in those superhero stuff, the mutations are great. They make you like powerful and stuff. But then here, the mutations are gonna make you sicker. So again, if you look at TMNT, just one turtle is not gonna do much. Like, I mean, Ralph could go in and Leah's gonna get stuck somewhere. It's not gonna be great. So the teamwork between all these four different like teams of mutant turtles makes a whole, whatever mission they accomplish better, right? So my research is on checking out if there's actually group of mutations that come in together rather than focusing on single mutations that actually can bring out much better explanation for how AD works. So I'm actually looking at what different types of mutations can I bring from the mothers and the fathers and how they influence the entire development. And it's actually a running research. And we have actually found from our School of Medicine's ADME program about 3 different sets of groups of mutations that actually influence AD pathology, but we don't really know how it works, but we know it is in the process. An interesting fact, it actually also influences cancer. So this was one of the the novel way of looking at diseases, and it's not just restricted for AD. And that is all my piece.
Great job. Thank you so much. All right, another quick pause for the cause. Also, you should have seen in the back a sheet that had graduate student resources on it. Please check that out. It has a number of resources there so that you can take advantage of those. It's the green one. Shumali, hold yours up. There it is. That's it. So find that sheet, it's in the back, and check that out. Also, we consider our graduate school website a one-stop shop for resources where you can find information on just about anything. So if you have a question about something, please go to the website, call us, let us know if you're having issues or have questions with some things, because the goal is to graduate, right? We want you to finish. Okay, we want you to finish too, I promise. That's right. All right, next we're going to have Vana, and Vana is going to talk to us about non-invasive classification of vocal pathologies.
innocently classify vocal pathologies. So first of all, just a background, approximately 1/5 of the world population suffers or has been suffering with the vocal pathologies. It could be due to aging or misuse of the voice, or even because they are just having some drugs or something like that. So how do we actually identify any vocal pathology? I know doctor would be the first answer, but also in this time of the health care and the technology, there are other methods as well. First of all, now like previously there have been subjective methods where you go to a doctor, they listen to you, and they rate to your voice according to the different rating scales. And also there's another method where they look at, they use the laryngoscopy, which is basically they insert a rod with a camera inside your mouth to identify what type of vocal pathology do you have. But then there comes my research, which is basically using the objective method, where they can identify your voice by using just the audio signal. So for that reason, I'm actually using the audio signals or the acoustic signals and the machine learning to classify between different type of vocal pathologies. But then comes the accuracy, like is it doing it accurately or not. In this case, if you see my results, the normal versus the pathology, I'm getting almost 100% accuracy. But if it comes to the two different classes of pathologies, I'm getting almost 95% accuracy. So yes, the future in this thing exists. We just have to come up with a best classifier. That's it. Thank you.
All right. Next, we're going to hear from Shweta, and Shweta's going to talk about reconcile, the broken synapses.
So good morning, everybody. Can you guys hear me right now?
Yes. Yeah.
So that's because me and you, we have our synapses intact. Synapses, they are the connections between our hearing cells and the neurons in our ears that help us to perceive this outer world. The synapses, they are like traffic signals controlling the perception of every outer auditory signal into our brains. And these, the hearing cells and the neurons in our ears, have a friendly relationship in such a way, in such a systematic fashion because that's how our mother nature is. It is the ultimate genius carving this marvel, our ear. But today, I'm here to tell you a story about baby Mia. Baby Mia, I met her 3 years back in the Riley Hospital. She was burdened with the heavy weight of the hearing aid, and I am trying to help her. How am I doing that? I hypothesize to utilize stem cells to study these synapses. Now, stem cells, they are like baby cells in our body that I can isolate and help them grow into these adult hearing cells and the neurons. And I could make the hearing cells and the neurons have a friendly relationship and a broken relationship. And that mimics the healthy and the broken, unhealthy state of the human beings in general. And that's how I try to utilize various drugs and try to mend or try to reconcile the broken synapses as per the friendly relationships of the natural state in humans. And that's how I am trying to help baby Mia restore her hearing ability. And I hope I can do this within my research time period here. Also, I would like to end my speech today by re-emphasizing on Albert Einstein's famous saying, once upon a time, he said, for us to delve deeper into the nature, to unravel the solutions to our problems. So that's how I would like to take a leave, and thank you.
Thank you, Shweta, and baby Mia. All right, so lastly we'll have Mauro, and Mauro's going to talk to us about reversing the battle of the gums in periodontitis.
Hello everyone. There is a battle going on right now inside the mouth of every one of us in this room. The battle is between cells that form the mucosal layer that cover our gums. On the other hand, we have pathogenic and invasive bacteria that is trying to survive and proliferate in plaque that cover part of our teeth. What's the commodity of this battle? What are they trying to win? They're trying to win gum, soft tissue, or bone, which is the hard tissue. Hard tissue is the alveolar, is also called alveolar bone, and it holds our teeth in place. In normal people, this battle is won by the cells that cover our mucosa. But in people that have underlying medical conditions or that drink alcohol or smoke, this battle is won by the bacteria. And what happened when the bacteria won the battle? Well, they eat the bone, they eat the gums, and now we are in something that is called severe periodontitis. Severe periodontitis is suffered for about 8% of Americans. And when you have periodontitis, your quality of life is diminished. And it takes about months or sometimes years to develop this disease. In the laboratory, we can mimic this disease by using rodents, and we put a silk suture around the second molar of these animals. We wait about 5 days, and at 5 days, we can see the same lesions that are characteristic of human periodontitis, but in the animal model. My lab is interested in studying a protein called PIC2. PIC2 is a protein that is present in different tissues, neurological tissue, cardiovascular tissue, and bone. In bone is very important because osteoclasts, that are the cells that eat bone, use this protein to achieve their function. So what are we trying to do in our lab is delete this protein, specifically in osteoclasts. And we hypothesize that if we delete this protein in osteoclasts, when we put a ligature around the second molar of mouse, we can prevent these animals from having bone resorption, and in this case, we are providing a tool for them to win the battle of the gums in periodontitis. Thank you.
Good job, Mauro. You really have me thinking about this battle going on in my mouth. Right? And I put candy on the table. Yeah, okay, it's my fault. So, our 3-minute pitch People's Choice Award goes to Natika. Natika. Yeah, come on. Natika, oh, sorry. Natika. Yes. Okay, so.
Yeah, can you get all of them?
This award comes with a prize of $200. Super excited about that. Love it. Awesome. Thank you.
All right, now we're going to start with our 3-minute pitch runner-up. Our 3-minute pitch runner-up is, drum roll please. It is Festus. Festus, come on up. Yes. Festus, you just won $200. taking us to all alpha tricks later.
Yay.
Yes, thank you.
Congratulations. All right, and our 3-minute pitch winner. And we usually send nice emails to your department, to your school, so that they know that you won. And we will highlight you in our Grad Jag newsletter. Yep, gotta love that. All right, are we ready? Drum roll, please. Yes, our winner is Natika.
Nithika, I'm sorry, I said that really wrong the whole time. Nithika. Yes. Congratulations. Love it.
Nithika just won $500 for 3 minutes of work. What? Yes. Love it, love it.
Well, I did want to say sadly that the rules are the rules, and particularly when it comes to these kinds of competitions, if any of you choose to go into this again, remember, no notes, no technology outside of your one slide. Anytime you violate the rules, you are instantly disqualified. And so unfortunately, we did have a couple of people who were disqualified for that reason.
Thank you for clarifying and for sharing that. All right. So now we're going to go to our People's Choice Award for our 3-minute thesis competition. Ooh, and I heard it was very close.
It was so hard.
Because like everybody did such a great job.
Stressed.
So yeah, so y'all stressed out my graduate assistant. Just letting you know that right now. And while I'm saying that too, let me give a great shout out to my grad assistant Charisma and to Lauren. Lauren started 2 days ago, y'all. Wow. So Lauren is being thrown into the fire. But we're really grateful to both of them for their help. Okay, so drum roll please. Our 3-minute thesis, People's Choice. Whoa. You voted, we heard it. It is for Shumalee.
Love it. Congratulations.
Great job, awesome, and thank you guys for voting. All right, next we're gonna go to our runner-up in the 3-minute thesis category. Now, this person, it's not like Miss America where
should they not be able to
perform their duties? No, but we will also highlight them and share about their research and let everybody know what an awesome job they're doing as well. All right, y'all ready? Drum roll. Ifama, let's go! Yes! You are runner-up. Yes! Super excited.
Love it. Great job. Love it. Congratulations.
We love it. Fantastic. And our Lily people are going to be super excited. All right. They are. All right. Now, for the moment we've all been waiting for. I know.
Our representative to the mags.
Yes.
Oh my goodness.
Our representative to Mags, who will carry our torch high.
Wow.
Who will represent us well. Our 3-minute thesis winner and the winner of $500 if they don't have a bill. All right. The bill will take your money, I promise. Okay. All right. Drum roll, please.
say thank you all again to all of our participants. You guys did an amazing job. It was fantastic.
It was great.
Our judges have commented on it, but people who were random that were in the room also commented on it as well. We're just like, they weren't random, they are the people we know. Also commented and were like amazed at it. We will post the video so you can share that with people and share it with your networks. And we thank you guys for attending today. Anything you all need, please remember that the Graduate School is a resource for you. And we are here to help you, okay? And thank you guys so much. I hope you guys add me to your LinkedIn network also. Bye, y'all. Have a great day.